Hello friends, welcome to BMH Learning. In this video, we are going to discuss about thin layer chromatography. Thin layer chromatography is used to separate different components from non-volatile mixtures. The experiment is conducted on a solid support which can be formed of aluminum foil sheet, plastic or glass. Onto these solid supports, a thin layer of adsorbent material is coated, which is nothing but the stationary phase. The material generally used to make stationary phase is aluminum oxide, cellulose, and mostly used one is silica gel. Once the separation is completed, each separated component looks like a spot separated vertically. Now, each spot has a certain retention factor which can be expressed in the form of following equation. The retention factor can be affected by a number of other factors. These are the solvent system, that is the type of solvent used. It also depends on the amount of material spotted. It also depends on the kind of adsorbent is used and also the temperature. Now let's talk about the principle of thin layer chromatography. Like other chromatographic techniques, TLC also relies on the separation principle. The idea of separation is that the different components of mixture has their own relative affinity towards both the phases, that is the stationary as well as mobile phase. When the components of the mixture are in mobile phase, they actually need to move over the surface of the stationary phase. So, in this way, the movement occurs in such a manner that the components having a higher affinity with respect to stationary phase move slowly. On the other hand, the rest of the compounds that have lower affinity to stationary phase, they will travel fast. So this is how the separation of different compounds is achieved. Once the separation process is completed, the individual compounds from the mixture are seen as spots. Next, we talk about the apparatus used to carry out the experiment. First thing we use is TLC plates. So generally we use ready-made plates. Special thing about these plates is that they are chemically inert and stable. Now, onto these plates is what we actually apply the stationary phase in the form of a thin layer. The stationary phase is formed of finely divided particle size and also the particles possess a uniform thickness. Second component is the TLC chamber. The TLC procedure requires a chamber in which the whole procedure would be carried out. Its function is to keep a steady environment that helps in developing spots. Other than this, it also prevents the solvent from getting evaporated. Plus, it also keeps the whole process dust-free. Third component is TLC Mobile Phase. So mobile phase will move, and through this phase, sample components would migrate. Mobile phase consists of a solvent or sometimes mixture of solvents. Now, this phase must be particulate-free. The more the quality and purity of the mobile phase, better is the development of spots. The fourth important part is filter paper. So the filter paper must be placed inside the TLC chamber. But why? We keep filter paper in the chamber so that equal humidity can be maintained. And this paper is moistened in the mobile phase itself. Now let's talk about the thin layer chromatography procedure. First, the stationary phase is applied to the plate and is kept to dry and stabilize. After that, we need to apply sample spots. So for this, with the help of a pencil, thin or light marks are made. But you should remember here, the marks must be made at the bottom of the plate. Then we need TLC chamber here. And after this, we pour the mobile phase into this chamber. And remember, to maintain equal humidity environment inside the chamber, we keep in the mobile phase a moistened filter paper. Next is the turn of TLC plate. Once everything is in place, the chamber is closed with a lid. The plate should be placed in such a way so that it can properly face the mobile phase. But remember, the sample spots on the plate should not touch the mobile phase. So we need to keep the sample spots well above the level of the mobile phase. Then we must wait until the spots are developed properly. Once the spot development is completely done, plate is taken out and is kept to dry. The plate with now developed spots is placed under a UV light chamber for observation. Next we talk about the applications of thin layer chromatography. It's used for the qualitative testing of a number of medicines like benzodiazepines, sedatives, anticonvulsant, tranquilizers, antihistamines, and steroids. Thin layer chromatography is very useful in biochemical analysis. That means it's used in the separation of biochemical metabolites from various body fluids. TLC can also be used for the identification of many natural products like essential oils, glycosides, alkaloids, waxes, etc. The technique is a very powerful tool for separation of multi-component pharmaceutical formulations. It is used for direct comparison between the adulterated sample and the original sample. It is also used in the food industry for the separation and identification of colors, sweetening agents, and preservatives. The method is helpful to study the process and completion of a chemical reaction. But unfortunately, the technique also comes with disadvantages. TLC plates don't have longer stationary phase. Length of separation is limited when compared to other chromatography techniques. It's difficult to reproduce the results generated from thin layer chromatography. Because TLC works in an open system, factors like humidity and temperature can negatively influence the final outcome. Detection resolution is low. Also, it's only a qualitative analysis method. This was all. Thanks for watching.